The title of my talk today is the regulation of pancreatic cancer-associated macrophages by CSF1 signaling pathway. So pancreatic cancer is one of the most aggressive forms of all malignant diseases, and the 5-year survival rate is less than 6%, and the median survival duration is only 6 months upon diagnosis. And current treatments are extremely ineffective, including surgical resection, which is only applicable to about 20% of the patients, and chemotherapy and radiation therapy are not being very effective at restraining tumor growth. However, the strategies involving the manipulation of the immune microenvironment offer significant potential in the development of effective anti-tumor therapeutics. So it's just beginning to be appreciated that the tumor microenvironment has many layers of immune suppression, which helps the tumor cells to escape from immune attack. And one of the major drivers of this immune suppression is macrophages, which are often one of the most abundant immune cells in the tumor tissue. And tumor-associated macrophages, or TAMs, are a very heterogeneous population and which contain multiple subsets that have different activation status and which and also carry on different functionalities. Based on their activation status, TAMs are often classified into M1 anti-tumor macrophages and M2 pro-tumor macrophage population. And these M1 and M2 populations usually coexist within the tissue microenvironment. Clinical data in resectable pancreatic cancer patients demonstrate that the patients who have high level of M2 and low level of M1 infiltration into the tumor tissue typically have significantly better prognosis compared to those who have low level of M1 and high level of M2 immune infiltration signature. So this makes tumor-associated macrophages a highly desirable therapeutic target. And the optimal strategy would be getting rid of this tumor-promoting macrophage population and enhance the activity of M1 anti-tumor macrophage population. How do we do that? The strategy that our lab has been exploring was by targeting the CSF1 signaling pathway. CSF1, also known as macrophage colony stimulating factor, is a cytokine that's essential for many aspects of macrophage activities, including their development, recruitment, and also survival. Compared to the cells in the normal human pancreas, human pancreatic cancer cells often upregulate the production of CSF1 ligand, which drives the infiltration of CSF1 receptor-positive macrophages into the tumor tissue. And we hypothesize that the blockade of CSF1 signaling pathway could enhance anti-tumor immune responses, therefore inhibit tumor growth. To address this hypothesis, we established orthotopic pancreatic tumor model by injecting syngeneic tumor cells into the mouse pancreas and treated these tumor-bearing mice with either neutralizing antibody against CSF1 ligand or a small molecule inhibitor that disrupts the kinase activity of CSF1 receptor. Using either approach, we were able to deplete about 50% of the macrophages that's infiltrating the tumor tissue. And correlating with this reduced macrophage number, CSF1 signal blockade also leads to significant reduction in both the primary tumor burden as well as the incidence of metastasis, which I'm now showing here. Now the question is, why are we seeing this phenotype? Does CSF1 signal blockade inhibit tumor growth simply because we have smaller quantity of tumor macrophages? Or is CSF1 signaling blockade also reprogramming the quality of macrophage responses in the tumor microenvironment? To understand the underlying mechanisms, we looked at the composition of tumor-associated macrophages. As I mentioned earlier, tumor-associated macrophages are very heterogeneous and may contain multiple subsets. And in the vehicle-treated mice, we were able to use CD126, which is a M2 macrophage marker, to identify a CD126 high M2 tumor-promoting macrophage population and a CD126 low subset that is enriched for M1 anti-tumor macrophage population. Upon the blockade of CSF1 signaling pathway, the majority of the CD126 high tumor-promoting macrophage populations are depleted, leaving behind these 206 low anti-tumor macrophages that dominate the macrophage composition. So this data suggests that CSF1 signal blockade preferentially depletes the tumor-promoting macrophage population, and also indicates that the remaining macrophages upon CSF1 signal blockade may have very different activities compared to the macrophages in vehicle-treated mice. So based on this data, we look at gene expression profile to compare the changes in macrophage functionality. What we did was using the same orthotopic tumor-bearing mice and treating them with anti-CSF1 or vehicle, and isolated the tumor-associated macrophages and compare the gene expression profile to look at the functionality on a per-cell basis. And interestingly, we found that CSF1 signal blockade significantly downregulates the immunosuppressive gene expression in the remaining macrophages, as shown in the red bars here. On the other hand, the anti-tumor immunity genes are significantly upregulated, and these genes are often correlating with enhanced anti-tumor T-cell responses. So what this data suggests is that the remaining macrophages upon CSF1 signal blockade may be less tumor-promoting and may have better ability to activate anti-tumor T-cell activities. So guided by this gene expression profile data, we performed a couple of ex vivo functionality assays, the T-cell suppression assay and antigen presentation assay. So for both assays, we isolated macrophages from anti-CSF1 or vehicle-treated tumor-bearing mice and looked at, in the ex vivo setting, how well these macrophages could either suppress or stimulate the activation of anti-tumor T-cell, CD8-positive T-cell activities, which is quantified on the y-axis here on both graphs. I'm not going to the details of these assays, but what we found was that macrophages isolated from vehicle-treated tumors, as shown on the white bars here, are very potent at suppressing the T-cell activation, and also very poor at presenting antigens to cytotoxic T cells. On the other hand, macrophages isolated from anti-CSF1 treated mice are much less immunosuppressive and significantly better at presenting tumor antigens to activate anti-tumor CD8-positive cytotoxic T cells. What this data suggests is that CSF1 signal blockade reprograms the macrophage responses away from the suppressive phenotype into a T-cell stimulating phenotype that better activates anti-tumor adaptive immune responses. And also consistent with this ex vivo data, we saw in vivo there is much less suppressive T cells and significantly more infiltration of anti-tumor CD8 and CD4 positive T cells. Taken together, this data suggests that blockade of CSF1 signaling pathway preferentially depletes the tumor-promoting macrophage population and allow the anti-tumor macrophage population to expand the presence of anti-tumor T cells. And next question we ask is, are these T cells functional? So T cell activities are regulated by a balance between stimulating and inhibitory signals. And the best characterized inhibitory signals come from a class of molecules called the immune checkpoint molecules, which include CTLA4 and PD1, which are expressed on T cells, and PDL1, which is expressed on cells that interact with the T cells, including macrophages or tumor cells. And upon the engagement of these immune checkpoint molecules, T cell activities are usually inhibited. And tumors often utilize these pathways to evade attacks from cytotoxic T cells. So we decided to look at the expression level of these immune checkpoints on both the tumor cells and tumor-infiltrating immune cells. And we found that upon CSF1 signal blockade, the expression of CTLA-4 on CD8-positive cytotoxic T cells is significantly upregulated. Also, interestingly, even though the tumor cells themselves express very minimal level of PD-L1, as shown on the blue bars here, CSF1 signal blockade dramatically upregulates their expression of PD-L1. So what this data means is that even though CSF1 signal blockade expands the presence of CD8-positive T cells, however, the engagement of these immune checkpoints, including PD-L1 on tumor cells and CTLA-4 on the tumor-infiltrating T cells, would make the T cells much less able to attack the tumor cells. And this may pose as a major limitation on the efficacy of CSF1 signal blockade. So to overcome this limitation, we thought, why don't we combine CSF1 signal blockade with immune checkpoint blockade? So we designed a combination therapy in our orthotopic tumor-bearing mice, and treating mice with both CSF1 receptor inhibitor and a combination of CTLA-4 and PD-1 antagonists. Blockade of CSF1 receptor signaling alone, as shown in the red bars here, or anti-CTLA-4, anti-PD-1 alone, have achieved modest effect at restraining tumor burden. However, when we combined CSF1 signal blockade with both immune checkpoint antagonists, we were able to see complete inhibition of tumor outgrowth and even significant level of tumor regression. So just to summarize the data I presented so far, CSF1 signaling drives the infiltration of tumor macrophages, which helps the tumor cells to evade cytotoxic T cell attack. And upon the blockade of CSF1 signaling, a subset of tumor-promoting M2 macrophages are preferentially depleted, leaving behind a population of M1 macrophages to expand the presence of anti-tumor T cells. However, CSF1 signaling also engages immune checkpoint, which include PD-L1 on tumor cells and CTLA-4 on tumor-infiltrating cytotoxic T cells, which limits the efficacy of CSF1 signal blockade. And therefore, we combine CSF1 signal blockade with immune checkpoint blockade, which achieved maximal, optimized ability of the immune system to inhibit tumor growth and ultimately leads to tumor regression. The combination of CSF1 receptor inhibitor with checkpoint inhibitor PD-L1 antibody has progressed into clinical trials in multiple types of cancer, including pancreatic cancer, and we are waiting to see its effects in human patients. And finally, I would like to thank members of our lab and collaborators and thesis committee and funding sources, and I would like to take any questions. We have still to see a bit of target effects in human patients. In mice, I would say the mice are very healthy. The major side effects would probably come from PD-1 antibody, which would restrain the efficacy of T cell responses in infectious disease. But macrophage-wise, you have overwhelming amount of macrophages that create immunosuppressive environment. So I would say CSF1 signal blockade will be more beneficial. Anyone else? Right. Thank you very much.